DNA, protein synthesis, genetic diversity, meiosis and selection, common exam questions part 2. This is for the AQA A-level Biology syllabus for the Unit 4 topic of variation. Aseptic techniques. Wash hands to remove or kill microbes or wear gloves to prevent contamination. Use a Bunsen burner close by to create upward current of air. Disinfect the bench or work on disinfected cloth to kill microbes or prevent contamination. Flame the equipment to sterilise microbes or prevent contamination. Lift the lid slightly to prevent entry of microbes. Directional selection. This is where one extreme is selected or favoured or chosen, or one extreme is allowed to breed. The genetic code is described as degenerate. This is because more than one codon codes for a single amino acid. Mutation may lead to still producing a functional protein. This is because a change at the amino acid site may not change the shape of the active site. A change at the amino acid does not change the tertiary structure. It could be an intron or a non-coding piece of DNA, so it's not translated and there's no change in the amino acid sequence. Explain how a mutation can result in the production of a non-functional protein receptor. A change in the DNA base sequence, so that's just a definition of a mutation. Change in the amino acid sequence or the primary structure of the protein. This alters the position of the hydrogen ionic or disulfide bonds. There is a change in the tertiary structure, which means there's a non-functional protein. Mutation may lead to a non-functional enzyme now, so looking at, in particular, at non-functional enzymes. So the first bits are similar. So there's a change or mutation in the base or nucleotide sequence of DNA. Again, just the definition of mutation. Change in the amino acid sequence or primary structure. There is a change in the hydrogen ionic or disulfide bonds. Therefore, there's a change in the tertiary structure. This changes the active site. Therefore, the substrate is no longer complementary or no enzyme substrate complexes can form. Chromosome non-disjunction. So in meiosis, homologous chromosomes or sister chromatids do not separate properly. This can cause an extra chromosome in the gamete or the egg or sperm that then formed a zygote, and because all cells are derived from the zygote by mitosis, all cells will have the extra chromosome. What is it meant by the fact that the genetic code is universal? In all organisms, the same triplet codes for the same amino acids. Genetic code uses 4 different DNA bases. What is the maximum number of different DNA triplets that can be made using these 4 bases? Well, there's 4 bases and a triplet is 3, so it's 64. Mutation leads to the production of a non-functional protein. Mutation changes triplets or codons after that point or causes a frame shift if it's a deletion mutation. This changes the amino acid sequence after this point. This affects the hydrogen ionic or sulfide bonds, changes the tertiary structure of the protein. Mutation. So, definition of a gene mutation, it's a change in the base sequence of DNA. This results in the formation of a new allele. It could be that mutation has no effect because the genetic code is degenerate, so amino acid sequences may not change. Or the mutation could be in an intron, so the amino acid sequence may not change. You can have a description of degenerate, so some amino acids have more than one triplet or codon. It might change the amino acid, but does not affect the tertiary structure because it's not where one of the bonds is formed. The new allele could be recessive, so it does not influence the phenotype. It could have a positive effect because it results in the change in a polypeptide that positively changes the property of the protein. This may result in increased reproductive success or may increase survival. Individuals in a population show phenotypic variation. What causes this variation? Could be due to genetic factors or environmental factors. Explain how variation is produced in crossing over. Sections of chromatids get exchanged. Sections have different alleles, so new combinations of linked alleles are formed. Natural selection. Variation is present in an original population. Individuals with the advantageous allele are more likely to survive. These reproduce and pass on their genes to the next generation, or to their offspring. There is an increase in the frequency of advantageous alleles or genes. Stabilizing selection. Stabilizing selection is natural selection that keeps allele frequencies relatively constant over generations. This means that things stay as they are unless there is a change in the environment. A classic example of stabilizing selection can be seen in human birth weights.